Well, good afternoon, everyone, and welcome to the Front Row. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research, and we're pleased to have our speaker, Martin Lotz, today. Before we get started, maybe I'll say a few things. First of all, it was great to see everybody sharing their locations. We have a global community today. I may have missed a few, but we have someone from Portugal, we've got Ireland, we have La Paz in Mexico, big crowd in San Diego, and it looks like some folks from Jupiter, Florida. So welcome, everyone. It's great to have you here. Now, I think the subject of today's lecture is something that's certainly on everyone's mind as we grow older. I have a feeling there are quite a few of us who have at least started to experience aches and pains from arthritis. And we're lucky to have today a real world expert on the subject of arthritis and developing new treatments for arthritis. And so that's Professor Martin Lotz. Martin is a professor in the Department of Molecular Medicine here at Scripps in La Jolla, California. Martin is an MD, and he got his medical training in Germany at the University of Heidelberg. And then he came here to Scripps to do a postdoc, and he did his clinical fellowship at UCSD. He's mounted a really impressive research program to try to really understand the molecular basis of the changes in our joints as we age and the basis for treating those kinds of aches and pains. So I think one other thing I just wanted to point out is that we had a bit of good news here last month. Since our last front row lecture in October, we were thrilled that Artem Panopinus won the Nobel Prize in Physiology and Medicine for his work on ion channels in the sensory neurons. So Artem actually gave one of the very first front rows that we ever had, maybe about 2.5 years ago. So I'd like to think we chose wisely. The Nobel Committee obviously chose, but it's a thrill for everyone here at Scripps to have his work recognized in this way. And I would point out this is really the first Nobel Prize where all of the research was done at Scripps. So we're very proud of that, and it's a special honor. So Martin, please welcome and take it away.
Thanks very much, Jamie, for this kind introduction. Yes, I can just only echo the comments you made about Scripps Research. It's an amazing environment, and actually it was for me impossible to leave. I came here as a postdoc, and I'm still here, and I'm still very excited to be here in such an amazing environment with such stellar colleagues, and also academic leadership, which is spearheaded by Jamie. Good afternoon, everyone, or good morning, or wherever you are. It is my pleasure today to introduce the topic of arthritis, and actually osteoarthritis in specific. So arthritis is a condition, actually a group of conditions that encompass two major forms, which is osteoarthritis and rheumatoid arthritis. The vast majority of cases with arthritis are actually patients with osteoarthritis, probably a ratio of about 10 to 1 or 10 to 2 compared to rheumatoid arthritis. Depending on the numbers, how numbers are calculated, there's approximately 60 million Americans that are affected by arthritis. And more than half of the individuals with symptomatic osteoarthritis are younger than 65. Osteoarthritis is an age-related disease, but it also affects individuals that are younger than 65 and maybe not consider themselves part of the aging population yet. The prevalence of arthritis actually is expected to increase in the next 20 years or so, almost doubling, and that's part of population aging. So the population that is at risk for developing osteoarthritis will actually increase, and also included in this projection is population growth. Another way to illustrate the current impact and also the greater impact of osteoarthritis in the future is to look at the number of joint replacements. The vast majority of joint replacements are for patients with osteoarthritis, where pain medications are no longer sufficient. And currently we have about 1 million such procedures done per year. And there is a staggering projection that this will increase actually to 4 million with only a time period of about 8 years from now. So that illustrates really the urgency for better managing this highly prevalent disease. So just to give you a brief overview of what I'm trying to convey to you today. So I will give you a brief introduction about disease impact and current status of mechanisms disease, how we do drug discovery, and then I will focus on 2 specific drug target candidates in the later part of the presentation. So osteoarthritis is a disease, not only a very prevalent disease, actually is a serious disease. It was long considered just something that as you get older, you have to put up with. But more careful examination has generated much detailed data about its overall impact. And this led the FDA to actually officially declare in 2018 osteoarthritis as a serious disease. And the reasons for this is that it's one of the leading causes of pain, second only to back pain. And it has tremendous impacts on the overall quality of life, sleep quality, mood quality. It's the third most rapidly rising condition that's causing disability. And it compromises the affected person's ability to self-manage other conditions that are comorbidities associated with osteoarthritis, such as hypertension, diabetes, obesity. Osteoarthritis actually increases the risk for developing heart disease by 50%, and it causes an overall increase in overall mortality by about 50%. So this illustrates just the tremendous impact and healthcare consequences of this condition. So what is osteoarthritis? What is happening in the joints that are affected by osteoarthritis? The key structural damage process occurs in the joint cartilage. The joint cartilage is this white tissue here that covers the ends of the bones in the joints, where 2 pieces of bones articulate with each other. It is the damage to this cartilage which is the driving cause of the subsequent involvement of all the other joint tissues. So once this process of cartilage damage, as shown here on the photos of the knees of the femoral condyles of a normal human and a 64-year-old with osteoarthritis, where you see that this tissue, the white cartilage tissue, actually has a different color in the older individuals. That's due to the age-related accumulations of changes in the collagen network. And actually here you see areas where cartilage is destroyed, illustrated schematically here. So once this cartilage destruction process proceeds, there is changes also in the bone that is underneath the cartilage. There's damage in the menisci, which are also very important in mechanical function of the knee. And there is also an inflammatory process that occurs in the joint and the synovial lining tissue, which is illustrated here, leading to joint swelling. And actually that's also a major manifestation, a major cause of joint pain. Just a couple more words about the joint cartilage. So the business of cartilage is to absorb mechanical load. When we are moving, our joints are carrying our body weight, and sometimes actually, for example, when we're doing a knee bend, 7 times our body weight. The cartilage is engineered to basically absorb mechanical load without wearing out under this mechanical pressure. So this is a histological image of the joint cartilage here. This is the superficial zone. Traditionally, the cartilage is just described based on the location of the area that you're looking at: superficial zone, mid zone, deep zone, and the calcified cartilage zone, which anchors the cartilage into the underlying bone. The interface between the cartilage surface and the synovial fluid is actually covered by a layer of proteins that are made up by an important molecule that's called lubricin. As the name implies, it's the main lubricant in the joint fluid that prevents wearing out of cartilage under mechanical load. The cartilage structure is made up by a collagen framework. Here you see the blue bars. These are illustrating the type 2 collagen fibers, and interspersed between the collagen fibers proteoglycans. Aggrecan is the major proteoglycan here. This is illustrated here by these yellow structures. This is the proteoglycan core protein, and these branches on the side, they're called glycosaminoglycans. These glycosaminoglycans are essential for biomechanical function of cartilage because they bind water, and water gives the ability of cartilage to actually deform under pressure, whereas the type 2 collagen is endowing cartilage with tensile strength and is actually the main determinant of the cartilage structure. The cartilage cell population actually is basically just one mature cell type, which are called chondrocytes. These cells are responsible basically for maintaining cartilage integrity throughout life. We've also learned that cartilage contains progenitor cells. So those are cells that are not yet mature chondrocytes, but actually have the potential to become mature chondrocytes, and actually, if abnormally programmed, could actually even turn into bone cells or fat cells. So they carry the signature of a progenitor cell. And that's an interesting population that we can actually also perhaps target in therapeutic approaches. Cartilage is a tissue that is not innervated. So the joint pain is actually not from nerve fibers and cartilage, but it's from nerve fibers in the joint capsule and in the meniscus. And there's also no blood supply. There's no blood vessels in articular cartilage. So it's a tissue that's functioning as a biomechanical shock absorber, and endowed, built by collagen fibers and aggrecan proteins with glycosaminoglycan side chains, and it's relatively homogeneous in terms of cell populations, the chondrocytes and some progenitor cells in there. So a current simplified view of how cartilage destruction occurs in osteoarthritis is that there are a number of risk factors that determine when and whether someone develops osteoarthritis. These so-called clinical risk factors include joint injury, they include obesity, which is a major risk factor of osteoarthritis, maybe malalignment, and of course, mechanical stress. So mechanical injury and mechanical overuse as a critical risk factor. And these metabolic diseases translate into biochemical stimuli that in the context of aging affect the chondrocyte behavior. So the cartilage cells actually do not proliferate during normal cartilage condition, and therefore are susceptible to accumulating age-related damage. What happens with these cells then is they are abnormally activated, and they produce mediators that cause damage to the extracellular matrix, to the collagen, to the proteoglycans, and also promote joint inflammation. So how is osteoarthritis being managed now? So what I'm illustrating here is a somewhat disappointing view when we compare treatment guidelines. Different organizations such as the American Academy of Orthopedic Surgeons, the American Rheumatology, they issue treatment guidelines. And actually those treatment guidelines haven't really changed very much since the time when I did my rheumatology fellowship and actually today. The only notable difference is actually the inclusion now of self-management programs, because it has been recognized that lifestyle, activity patterns, types of physical activity can actually have an impact on disease progression. So the types of physical activity can actually make disease worse, or they can actually make disease better, leading to improvements in pain and also potentially slow down progression of the disease. So the fact that we don't have any pharmacological treatment that can stop progression or even prevent the onset of disease is not because there haven't been any attempts at developing such drugs. Such drugs we call disease-modifying osteoarthritis drugs. And there have been a few hundred drugs that have been tested. And so far, every single one of these clinical trials has actually failed to show efficacy in patients with osteoarthritis. And this is just trying to give you a flavor of the types of drugs, the types of drug targets that were pursued in osteoarthritis drug development. So the first wave of clinical trials were actually testing inhibitors of the enzymes that break down collagens and other cartilage structural proteins. There was a wave where drugs that have effects on bone metabolism were tested, inflammatory mediators with interleukin-1 receptor antagonist, inos inhibitors. Most recently, cellular senescence is a key process that is targeted in many diseases, including osteoarthritis, but also this senolytic drug failed, and another inhibitor of an enzyme that breaks down cartilage has failed. So why is it that these clinical trials have not been able to show significant improvement in patients' disease onset or progression? So this is my view of the landscape of clinical trials in osteoarthritis. And I think there are critical barriers in clinical trials design. And that includes, first of all, patient heterogeneity. So the patient population with osteoarthritis is very diverse in terms of their risk factor profile, in terms of their activity profile. In clinical trials, most of the patient populations that were enrolled were actually patients with advanced stages of disease, relatively high average age. And what we've learned over years of research in our laboratory is that when we actually look at patients who undergo joint replacement surgery and examine the cartilage that is being removed by the surgeons, almost all of those samples actually have abnormal calcification and also amyloid deposits. Amyloid deposits are a key driver of many age-related diseases, including dementias, but they're also very prevalent in disease. And in fact, in osteoarthritis, this integration of this knowledge, this translation of this knowledge into clinical trials design has not happened. There is no consideration, in fact, of whether or not a patient has these abnormal age-related deposits. And we've actually done studies and shown that amyloid is not just an innocent bystander, but actually can promote disease progression. Also, some patients have a high load of clinical risk factors. So an older individual with obesity, overuse, or injury is at high risk for not only osteoarthritis onset, but also at high risk for progression. And I think there is probably subsets of this population that are beyond the opportunity for a drug intervention to actually have a benefit. Probably in those individuals addressing these risk factors and then perhaps adding a drug may be an opportunity. So there are major issues in clinical trials design in osteoarthritis. Another main reason, of course, is that the drugs that were tested may have hit the wrong target. And that's, I would posit, certainly true for the first wave of drugs that were tested. So taking the example of the breakdown of collagens and the other structural elements of cartilage, there are probably 40 or more enzymes that are at play to destroy the joint cartilage. And these trials actually have just picked one out of those many enzymes and made inhibitors for them. And they didn't succeed in clinical trials. The same applies for inflammatory mediators. There's a whole array of inflammatory mediators. And targeting a single mediator probably is not going to be efficacious. Furthermore, we now have developed a better understanding of osteoarthritis pathogenesis, mechanisms that cause the disease. And we know that there are multiple pathways at play. And how to determine which of the pathways is the key driver of disease and how to target them is another important challenge. So that's where the aspect of drug targets is really essential. So what we need is a better understanding of disease mechanisms and then prioritize and identify the key drivers of tissue damage and pain. And that would, of course, then translate into better drug targets. So that's the area in which our laboratory is working. So we are trying to better characterize mechanisms of disease, not just for the basic understanding, but directly then acting on them as drug targets. So in the next section, I will address how we actually go about drug target discovery in osteoarthritis. And the approach that we are currently using is a technology-driven approach. In the last 5 to 10 years, we have very much benefited from technical advances in sequencing DNA, in sequencing the RNA molecules, the messengers for protein synthesis, and also sequencing other modifications in DNA that determine whether a gene is actively transcribed or not. And more recently, we are benefiting from the advent of a new technology to actually look at all of these changes in nucleic acids at the single cell level. So this technology-driven advance is now giving us the opportunity to characterize the transcriptomic and epigenomic landscape of normal versus arthritic cartilage. And we're also looking at meniscus to identify pathways that are central to the pathophysiology, and then use this data to actually identify therapeutic targets, and among those, select those that seem to be most promising. In our transcriptomic analysis, we have a specific focus on a particular set of factors that are called transcription factors. The function of these transcription factors is that they basically turn on or turn off the transcription of a gene. So these factors decide whether or not we generate messenger RNA. And they are also critical determinants of tissue identity and regulate the signature genes that make cartilage and meniscus. And it is a hypothesis that dysregulated transcription factors control the expression of many disease-promoting genes, not just one inflammatory mediator or one of the many enzymes that break down cartilage structure, but regulating whole sets of genes, and therefore being more potent when we overexpress or inhibit them in therapeutic approaches. So this is an example of just sequencing the RNA molecules that are expressed, the messenger RNA molecules that are expressed in joint cartilage. So we're taking cartilage tissue from normal individuals, from tissue banks, and from osteoarthritic individuals that have joint replacement surgery. We isolate the RNA there, and then perform genome-wide sequencing. So we actually have a global genome-wide information about which genes are up or down regulated. What this graph shows is that actually we can differentiate normal cartilage from osteoarthritic cartilage by these gene expression patterns. So the osteoarthritics in red and the normal samples are clearly separated. What determines the separation is that of the approximately 12,500 genes that are expressed, about a little over 10% of these are abnormally expressed in joint cartilage of osteoarthritis patients, and about 630 of them are abnormally high, and about 700 of those are abnormally low in osteoarthritic cartilage compared to normal cartilage. So to make sense out of these sets of genes that are abnormal, we then asked, following our theme of transcription factors, how many of these 1,300 abnormally expressed genes are actually transcription factors. And so there's 93 of the transcription factors that are abnormally expressed in osteoarthritis. And actually what was quite striking is that a large majority of these factors are actually suppressed. So then we integrated the information about the transcription factors and looked at all the entire gene set that is abnormal in disease, and asked which of these abnormally expressed transcription factors actually control the expression of these differentially expressed genes. So targeting differentially expressed genes, meaning those transcription factors would be the switches that turn on or off these genes. And what came out of this analysis was a class of transcription factors that called Foxo. So these Foxo transcription factors, they were actually among those suppressed factors in osteoarthritis. But not only that, these transcription factors actually hit and control expression of many of the genes among these 1,300 that are abnormal in disease. So those transcription factors, obviously, with that property, look promising as important player in disease. This is looking at abnormal gene expression actually at the single cell level. So this is what's called single cell RNA sequencing. So what we're doing here is we are actually isolating the cartilage or meniscus cells and then labeling them and put them through a machine where single cells are labeled, and then we can actually do sequencing of the genes that are expressed in each and every single cell. Then we take the sequencing information and actually reconstitute cell populations that have similar patterns of gene expression. And when we do this for cartilage and meniscus, we have somewhere around 15 to 20 different cell populations that constitute these two tissues. Then we, of course, ask the question, which of these cell population is enriched in genes that promote disease progression? This analysis in each of the tissues in the articular cartilage and in the meniscus actually identified what we call pathogenic subsets of cells. So those are the genes that are very enriched in producing many of the inflammatory mediators, many of the tissue-destroying enzymes. So we have one of these populations here in osteoarthritic cartilage that is enriched in osteoarthritis promoting genes, and the same is true here for meniscus. And actually, both of these cell populations are deficient in Foxo. So this Foxo deficiency is seen at the whole tissue level in RNA sequencing. And this point here illustrates that it's actually very deficient in especially those cells that appear to be key drivers of the disease process. So a brief summary here about this RNA sequencing studies. So osteoarthritic cartilage has a high expression of genes that are involved in the extracellular matrix, which is the cartilage structure molecules, and the extracellular matrix remodeling, and reduced expression of genes that are beneficial, that promote what we call tissue homeostasis. Among those, the Foxo transcription factors. So based on these findings, our hypothesis is that Foxo deficiency is a mechanism for disease-promoting gene expression patterns in osteoarthritis cartilage. So I will now provide you a summary of our work on these Foxo transcription factors. So Foxo transcription factors, of course, have been well studied in other contexts. And perhaps the most important biological function that has been identified is that they have very essential protective functions in cellular health. Foxo are critical determinants in determining lifespan, in determining cell health, cellular health over the course of a cell's life or a tissue's life. One of the processes that is closely regulated by Foxo is one of these cellular homeostasis or housekeeping mechanisms called autophagy. So that's a mechanism where the cell actually has a mechanism to recognize which of its constituents are damaged. For example, the cell can specifically recognize when there's damage to the mitochondria, the powerhouses of the cells. And when that is recognized, then this self-eating autophagy machinery gets turned on, and actually the damaged mitochondria are engulfed in what's called an autophagosome, and they're broken down into their building blocks, which then are actually used for recycling. So this is a fundamental mechanism that is controlled by Foxo. So not only because of differential expression of Foxo, but also because of the importance, the known importance of Foxo in determining cellular health during aging. They are promising targets for osteoarthritis. So this is just introducing an animal model where we also confirmed that not only in humans, but also in this animal model, we actually have in the function of advanced age, a suppression or loss of Foxo expression. So this is just what we call a wild type mouse without any genetic modification. And mice, as they get older, also develop age-dependent osteoarthritis. And that is associated with a reduction in Foxo1 and Foxo3 expression. What's shown here is also another way we induce osteoarthritis in animals, which is actually surgically, where we cut out a piece of the meniscus or destabilize the meniscus surgically, and then mice develop osteoarthritis in a short 2-month time period. And in this animal model, we also have a suppression of Foxo. So there's a very close correlation between osteoarthritis progression and loss of Foxo. So then we next studied the function of Foxo in live mice. So the approach here is to actually use a genetic deletion of Foxo. And when we did this in mice that had the Foxo deletion already during fetal development, we found that these mice, when they just became skeletally mature, actually had severely damaged cartilage, and by 6 months old, they actually had complete loss of joint cartilage. So that finding demonstrated that Foxes are really playing an essential function in cartilage development and maturation. And without Foxo, the cartilage will actually no longer, will not sustain. The limitation in this experiment is that it actually leads to a developmental defect, whereas osteoarthritis, of course, is a disease of mature individuals. So we used a different genetic approach and turned off the Foxo genes in mice only after they were skeletally mature, which is by the age of 4 months. So what we can do in these engineered mice is we can engineer the Foxo gene such that it will be only deleted in cartilage because we're using this aggregant driver, and it will only be deleted if we give the mice a drug, tamoxifen. So that's been our approach here. And then once these mice had Foxo deletion, we looked at gene expression patterns whether they develop osteoarthritis under different circumstances. So deletion of Foxo in mature mice also leads to osteoarthritis. So what happened here is mice were treated with tamoxifen to delete Foxo at 4 months, and by 9 months they actually had severe osteoarthritis. So this is here quantified. This is a scoring for the severity of cartilage damage, meniscus damage, and synovial inflammation. And you can see here the 3 red groups of mice with the Foxo deletion have more severe disease. And this is associated with deficiency of genes that control this homeostasis mechanism, autophagy, and protect cells against antioxidants. So the gene knockouts lead to a reduction of all of these genes. When we do the converse experiment and actually take human chondrocytes and overexpress the Foxo gene by putting in a virus that carries the Foxo gene, then actually these same genes that are deficient in the knockout mouse are actually increased in the human cells. Another important function in Foxo, and a very essential function, is that it controls the expression of this protein called PRG4, or lubricin, the main lubricant molecule in the joint fluid. So again, here, this molecule is deficient in the Foxo knockout mice. And when we take the Foxo gene and express it in different human chondrocytes or mouse chondrocytes, then Foxo increases the expression of PRG4. A coincidental observation in this experiment was that when we knocked out the Foxo genes, they not only had more severe osteoarthritis, but they actually had overt spine abnormalities, and leading actually to a severe kyphosis. And then histologically, we can see that there's abnormal formation of the intervertebral disc and degeneration of the intervertebral disc. So there's many similarities in mechanisms of spine degeneration, the intervertebral disc degeneration, and knee osteoarthritis. So we actually have several parallel projects where we were studying similar mechanisms in both tissues, in both disease contexts. And we now have a program where we're also examining Foxo in intervertebral disc. So to summarize those findings from the Foxo knockout mice, they develop damage to cartilage, meniscus, and intervertebral disc. Foxo controls these important protective function, enhances the expression of this joint lubricant. When we take osteoarthritic chondrocytes and put a virus that carries Foxo1 gene into human osteoarthritic chondrocytes, then we normalize gene expression. The Foxo deficient mice not only have osteoarthritis, but also damage to meniscus and intervertebral disc. So collectively, these findings classify Foxo as a promising therapeutic target for osteoarthritis and intervertebral disc degeneration. So to then actually act on Foxo as therapeutic targets, we set out to discover small molecules, drug molecules that would actually increase the expression of Foxo, because in osteoarthritis in humans and mice, there is a suppression, a reduced level of Foxo. The way this is done, we used a piece of the Foxo promoter, that's the part of the Foxo gene that determines whether the Foxo DNA is transcribed into RNA. And then made what we call reporter constructs, which give us a light signal if a small molecule binds to the promoter and activates it. So that's our reporter assay. And the compounds that we tested in the screen are from a selection of about 13,000 compounds that was created by Caliber, which is the drug discovery and drug development branch of Scripps Research. So what Caliber did is actually collected these drugs that either are approved drugs or have been actually in advanced clinical trials for the potential for repurposing. Obviously, these drugs all have extensive preclinical information. They also have information about clinical effects, about potential toxicity, about potential mechanisms. So this is in a way a gold mine for drug discovery, for repurposing. So performing the screen with approximately 12,000 compounds, we identified 113 so-called hits, which gave it signal in the reporter activating the Foxo gene. And then we used several criteria to narrow down this list to about 18. And then we took these 18 candidates and tested them in joint cells. And with that, again, we were able to rank them in terms of the extent of their protective or beneficial effects. And the top compound that came out of this approach is a compound that's called Ponobinostat. And that was what was then tested in an animal model. So Ponobinostat is a compound that inhibits histone deacetylases, which are important regulators in transcription and also in protein modification. And the inhibitor that we tested, Ponobinostat, has also important activity against this particular class 2 HDACs, and of all of the other pan-HDAC inhibitors, Ponobinostat had the most promising profile. So that's why we proceeded with more detailed testing of Ponobinostat. Here in cartilage cells, we show that it inhibits the expression of inflammatory mediators and enzymes that break down cartilage. When we inject it in the mouse joint, it has a similar activity in reducing this enzyme that breaks down collagen and also this inflammatory mediator. So then we used the osteoarthritis animal model in mice, where we destabilize the meniscus, and then within a time frame of 8 weeks, these mice develop osteoarthritis. And upon treatment with Ponobinostat, we see a dose-dependent improvement in cartilage damage. And what we also can do in mice, we can actually test pain behaviors. And that's done by measuring pain threshold, the sensitivity to pain, which in mice with osteoarthritis, of course, there's an increased sensitivity. So this reduction here is a surrogate for reduced pain sensitivity. So to summarize these studies, we have from this free-frame drug screening identified these HDAC inhibitors and Ponobinostat as the most promising compound in cell assays, and they also, Ponobinostat also is efficacious in an experimental osteoarthritis model, where it reduces the severity of tissue damage and also pain behaviors. So having both of these properties, having a benefit on pain and inflammation, but also on structural disease progression, this has a clinically relevant activity profile and is a candidate for osteoarthritis symptom and structure modification. So in the last part of my talk, I will discuss an approach to address damage to the meniscus. So the knee menisci are also essential for biomechanical function of the joint. There is a high prevalence of meniscus damage with aging. So this is actually images of human knee menisci. This is what a normal meniscus looks like. And here you have different degrees of degradation. And if you're looking at this advanced stage, you see here you have major pieces of the tissue have been broken down. You can actually see here this white material is macroscopically evident calcification, illustrating how prevalent this age-related modification is. So meniscus damage occurs either after acute trauma, like when a sports injury is associated with meniscus, with ligament tear. The ACL tear is most of the time associated with meniscus as well. But with aging, there is a so-called development of so-called degenerative meniscus tears, and that accounts for the high prevalence of meniscus tears in the older population. So what we set out to do is to, again, ask what are the transcription factors in meniscus that actually identify the meniscus cell phenotype, and can we use those to address meniscus damage? So here we took advantage of a large National Institutes of Health endeavor, which has sequenced about 40 human tissues. And we can then use this information about gene expression patterns and ask which are the transcription factors that are most enriched, in this case, in meniscus. And so this gene expression analysis identified a transcription factor called MOHOC that is most enriched in meniscus. And even comparing it to cartilage, there's a big difference in the expression of MOHOC in cartilage. So MOHOC actually is a factor that can turn a stem cell into a meniscus cell, indicating that it's a very important mediator of identifying and determining the phenotype, the signature gene expression that makes a cell a meniscus cell. And this is just illustrating some of the genes that in fact make determine the phenotype of meniscus cells. And this is images of these pellet cultures of stem cells. And when we combine MOHOC gene transfer with this growth factor TGF-beta, we have a high expression of type 1 collagen, some expression of type 2, and expression of aggrecan. So we can turn a stem cell using MOHOC into a meniscus cell. And when we look at actually mature meniscus cells, we're taking cells out of meniscus and putting MOHOC in there, then we see that actually it inhibits many of these genes that are involved in meniscus damage and also in the abnormal meniscus calcification. Then we took human meniscus tissue that was removed during joint replacement surgery and injected virus that leads to expression of MOHOC in the meniscus, and then looked for the potential of MOHOC to actually cause repair of tissue damage in osteoarthritic menisci. And that's what we're showing here. With the addition of MOHOC, we actually have more collagen formation and glycosaminoglycan deposition. And we're using a scoring system for the degree of meniscus damage. That scoring goes down, meaning an improved tissue architecture with viral Mohawk gene transfer. And that's associated with, again, inhibition of all of these mediators of meniscus calcification and meniscus damage. Then we use the gene therapy approach in the animal model. So here, meniscus was destabilized surgically, and that leads, as shown, this triangular structure is the meniscus, and that leads, the surgical procedure leads to severe meniscus damage. And then we injected the virus carrying the Mohawk gene into these knee joints, and then performed scoring for meniscus damage, for cartilage damage, and for synovitis. And for all outcomes measures, we saw significant improvement after viral Mohawk gene transfer. So the summary of our Mohawk studies is that this is the most enriched and important transcription factor that determines meniscus cell identity. In menisci from human osteoarthritic joints, Mohawk gene transfer promotes repair and suppresses genes that are associated with meniscus calcification and structural damage. And in a mouse model of meniscus injury, the injection of Mohawk carrying virus into the joint leads to healing of meniscus tears and actually reduces the associated cartilage damage as well. So we conclude that meniscus gene delivery is a promising approach to prevent osteoarthritis, post-traumatic osteoarthritis, which is associated or a consequence of meniscus damage and slows the progression of age-related disease. So where we are currently headed is for small molecules such as Ponobinostat, develop local therapies for injections into the joints and into the intervertebral discs, so that we can actually have formulations where a single injection every 3 months or so is sufficient to have sustained drug delivery. And the other approach, specifically for the transcription factors, would be to pursue local virus-mediated gene delivery for Mohawk or other transcription factors that we are working on. Last but not least, and most importantly, I have to express my gratitude for my team that has made all of this work possible. So there are former and some and all of our current lab members that have contributed to these various projects. I want to point out my long-term collaborators, Hiroshi Asahara and Daryl De Lima. I also want to thank very much Marissa Olmer, who is an expert in many laboratory techniques and a lab manager as well. And Connie Choi, who is a lab coordinator and keeps us organized and sane. And I also have to acknowledge, I want to say special thanks to Kristen Johnson, who allowed me to work with Caliber on one of their clinical stage projects and also for the drug discovery here. And our funding comes from the National Institutes of Health, and specifically, actually, all of our funding now is from the National Institute on Aging. Last, I want to let you know about the upcoming lecture. Next month on the 15th, we have a presentation from the Scripps Science and Translational Institute about their approach to use digital, to establish a digital clinical trials center, and how this is enabling a completely new approach to engaging a large segment of the population in clinical research. With that, I thank you very much for your attention. And of course, I will be happy to answer any questions.
Thanks, Martin. Terrific lecture. Lots of interest where questions are pouring in. I'm going to try to kind of aggregate a couple of the questions. So one thing that people were kind of catching on to was this idea that there might be oxidative stress or an increase in autophagy related to inflammation and a link to osteoarthritis. So can other ways of reducing that sort of impact osteoarthritis, and what's the connection between, let's say Foxo and all of the above?
Yes, there are many entry points into blocking these pathways that cause disease onset or progression. So, for example, mitochondria are also an important player in all age-related diseases, and also specifically in osteoarthritis. There's actually increased accumulation of damaged mitochondria in osteoarthritis cartilage. And those damaged mitochondria, they are actually the source of increased oxidative stress. So one way of trying to address this would actually be to develop drugs or approaches that would correct the abnormalities in mitochondria. Another way would be to actually use drugs that sponge up oxygen radicals. But in my concept, I don't think this would be by itself sufficient, because there's just so much else happening, so that we need multi-pronged approaches to control the overall drivers of disease process. And the other connection that's important here with Foxo is that Foxo controls the major mechanism that actually removes damaged mitochondria. It's a special form of autophagy. It's called actually mitophagy, which specifically recognizes mitochondria and removes and recycles them.
So another kind of theme is running through. You mentioned a couple times tamoxifen, and I think there's people taking tamoxifen, and they're a little interested. It suppresses Foxo, but would that actually increase their arthritis? And how do you unpack all of that?
Let's forget this question.
Okay.
This is just used as a research tool to turn on the engineered gene to be either overexpressed or knocked out. So it's actually not an inhibitor of Foxo.
Right.
It's just a tool in the mouse, in the genetic mouse models.
Yep. Thanks for that. Yeah. And I think a number of people also ask, can you do gene therapy, or can you just inject Foxo? But my understanding would be that's a transcription factor. It's a protein, and that's not what you need. What you need is a way to control its levels with a medicine.
Yes, so for Foxo, in fact, that's why we used the small molecule discovery as a way to increase then the transcription of the Foxo gene, increased messenger RNA, leading to increased Foxo protein expression. Right. We're pursuing another approach for Foxo, which is actually once the Foxo protein is synthesized, it then undergoes a series of further modifications that are called post-translational modifications that actually then control the activity of the protein. And there is, in the context of other age-related diseases already a substantial effort to discover Foxo protein activators. Now, in the case of Mohawk, the transcription factor that is deficient in disease and controls meniscus regeneration, there we actually think that gene transfer would be a viable approach. So we can actually, like we've done in the mouse model, we can inject a virus that carries the Mohawk gene into the joint, and the virus then infects cells in the joint tissues and leads to increased Mohawk expression. And in the mouse model where we caused meniscus damage, this actually led to meniscus repair. And we were worried whether this would actually be causing side effects, for example, by infecting cells in the joint capsule or in the synovium. And we actually didn't see any side effects there.
Right. So a couple of related questions. Are there any known disease mutations in Foxo And are there just other genetic risk factors? So if you have your genome sequenced, and is there anything to look for, or what can we say about human genetics in this disease?
Perhaps the most striking observation about Foxo and genetics is there are genetic polymorphisms in the Foxo3 gene that correlate with longevity. And actually, this Foxo polymorphism is one of the two most powerful genetic polymorphisms having an effect on human lifespan. And now, concerning Foxo polymorphisms in osteoarthritis, there is no evidence for a genetic risk. Mm-hmm. Actually, a very recent study, the most comprehensive study about genetic association with osteoarthritis, looked at more than 800,000 individuals, and they identified about 100 gene polymorphisms, where each one of them had a small effect on increasing risk for osteoarthritis. But Foxo polymorphisms were not included there.
We got a bunch of questions about Ponobinostat. People are lined up for signing up for clinical trials. And I think I just want to remind everyone that this drug came out of a library that Caliber has of all known drugs. So yes. This is somebody else's drug for something else, and you found that it affected this purpose. So what's the path forward now that you know you've got a compound that does this to make an OA medicine?
Yeah, right. So first of all, this is the purpose of drug repurposing.
Yep.
So actually having a drug that's, in fact, Ponobinostat is clinically approved for the treatment of cancer. Mm-hmm. And so we already have a huge database of information about this drug. The path forward then has a few challenges. Number one is it's someone else's drug, so you would have to reformulate it, and our solution to that would be actually to develop a sustained release formulation, so we can actually inject the drug into the joints so it acts locally. So that would give us a potential for creating intellectual property. And so that would be our path forward for Ponobinostat. We would not advocate oral or systemic administration of the drug. We would actually develop it for local injection. Now, in osteoarthritis, there in fact is an opportunity for local treatment because, for example, most patients with knee osteoarthritis really just have osteoarthritis in that one knee that's really bad. So there we have feasibility for a local approach. So then the next hurdle is to convince investors to support, or now we actually have more funding from the NIH, which would be perhaps more readily available to actually do clinical testing of such a formulation.
Well, thanks a lot, Martin. I have one more question for you, but I just really want to add a comment to emphasize to the audience how hard this really is. And you're really making groundbreaking insights into the mechanism of osteoarthritis, yet we have to acknowledge we're years from realizing that as treatment. And that's our role at Scripps Research, is to find the next generations of medicine. And so you've got a remarkable program here. It's wonderful to see everything coming out. Now, so I just want to close with just a personal question. Martin, how did you come to be in research? You went to medical school and somehow you ended up in the lab. And so, what were the key points in your life that sent you down this road to Scripps?
So I think, first of all, a key driver for what I've done professionally in my life in general is that I have a limitless curiosity. And I think my curiosity was channeled by important influences in my environment growing up. So I like to understand how things work, and I actually also have sort of an intrinsic desire to fix things that are broken. And I actually cannot imagine a better environment for that than the environment that I grew up in, which was a farm in Germany. And on a farm, you have fauna, you have flora, you have machines that break down, and you have animals that are sick, and you have crop that doesn't thrive. So it was really an amazing laboratory in a way. But then I have to acknowledge two really important mentors. One of them was my biology teacher, who made me embark on my first research project when I was 16. And then I have to, when I came here to Scripps, acknowledge Dennis Carson as my mentor. And he is, I think, a stellar role model for a translational scientist. And then, I think it's just been no better environment possible for me to imagine than the environment that I've been finding here and thriving in here at Scripps.
We at the end of the day, we have two products. We produce science and important results that will impact human health. And in doing that science, we train the next generation of scientists. So that you're now mentoring those next people, and hopefully will have an equal impact as they had on you. So mentorship is a really important part of what we do here at Scripps.
Yeah, and it's an amazing opportunity for us to share knowledge and actually also see how we can shape others' careers. Yes.
Okay, well, thanks, Martin. Thank you very much. And then again, to remind everyone, December 15th, we have Katie Bakamotis and Julia Moore-Vogel, and we'll be hearing about digital trials. So I expect to see all you at the front row. Bye, everybody.